Hello, my name is Omar Kana, and I will be presenting on our poster, Single-Cell Universal Differential Equations, a machine learning framework for extracting ordinary differential equations for gene regulatory network inference. Underlying all developmental trajectories are representative expression pseudotimes, which represent a single-cell ordering of cells along the developmental trajectory of interest. We utilize pseudotime inference in order to infer values of developmental time along these trajectories and utilize that to graph the regulatory cascade of gene biomarkers as well as patterns of co-expression along the cellular lineage. In this way, we utilize this information to infer interactions between genes and thus construct a gene regulatory network. To infer the nonlinear interactions between genes, we utilize the Cindy algorithm. In this case, we look for a basis xi over a candidate function of a library of functions theta on x on our expression matrix. This theta on x can represent any set of functions selected by the user. In this case, we look only at biologically relevant functions like the linear function, the exponential function, and Hill functions on the expression matrix. We then look for xi using lasso regression. And thus our representative xi represents our gene regulatory network. Unfortunately, Cindy requires a time derivative for the system. In this case, we utilized a neural ordinary differential equations to derive the time derivative of the system. To validate our model, we use a simple bifurcation model from the Boole ODE framework. In this case, we utilize slingshot in order to infer the pseudotime trajectory of our model. Then we utilize the neural ordinary differential equations framework to derive the time derivative of the system. And then we infer the symbolic representation of our system using the Cindy algorithm. In this case, we wanted to see whether or not the Cindy algorithm inferred symbolic system is actually nonlinear. In this case, we looked at several different initial conditions, which produced two distinct trajectories within our system, in which we have two examples represented in our UMAP trajectory on the right. We then validated our method on a more complex biologically based method from the Boole ODE framework based on synthetic myelopoiesis. In this case, we show that we both can reflect the expression trajectory of the data as well as outperform other methods in the literature on the network inference task based on the area under the precision recall curve. Finally, we want to validate our network on real hematopoiesis data, which we've taken from a mouse hematopoiesis data set by Nestorova et al. In this case, we looked for the top 10 highly variable transcription factors in our data set and constructed a true underlying network using public data sets listed below. We then validated our method in the same way that we validated on the synthetic myelopoiesis data set on the network inference task. As shown here, we show that our algorithm can outperform the other two ODE-based algorithms, SCODE and Grizzly, on the network inference task. In green represents true positives, while in red we see false positives. In conclusion, we showed that SCODE can infer complex nonlinear dynamics in these models and systems. We also show that SCODE can outperform other ODE-based models and the network inference task in both synthetic and real hematopoiesis datasets. SCODE has its limitations. However, we discuss different ways that those limitations can be rectified in the poster presentation.